Hello. Very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible to you, and also if you can see the presentation? Let me know in the chat box if my voice is audible and also if the PPT is visible to you. Okay, thank you. We will wait till it's 6 p.m. and then we will start today's session. Okay, so it's 6 p.m. I think I will wait for 2 to 3 more minutes and then I will start the session for today. Okay, let's wait for a few more time, then I will start the session. Okay, so I think I should start the session for today. So let me just check. Okay, so I will start the session. Meanwhile, when other students will join, I will just they can just join because I have made the call open right now. So very good evening to all of you. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry. Today it is our week 12 live session. So today it is our last live session for this course of Medicinal Chemistry. From past 11 weeks we have solved different kind of questions related about different topics of medicinal chemistry, and that's how we followed the session. So today in our last class, so right now what we had do in past previous 11 weeks, that whichever week is live that time I have taken 20 questions from that and I have shown you have solved it correctly, and also you have tried very well to solve the questions, and then I have shown you the answers and we have proceeded like this. And but today it is our last live session, so we have already completed our course, and also we had our summary session number 1, which was on last till on the last Sunday on 14th April. So we had our summary session also. So in our today's session what I will do is, I will again, it is kind of a summary session but it will have few questions at last. Okay, after the summarization part I will have few questions for you to check if you have prepared for your exams or not. So it will like a kind of a summary session along with a question answer session. So I will give you the question, and the questions will be from your all the topics which are in the particular course, like your total 12 weeks I will have I think 10 to something questions from the all the questions. But before going to the question answer session I will start with summarizing few points. Okay, so these topics I thought it were in your course all, but I thought it will be better if I can give you a little bit more about the topics, so it will be helpful for you, not only for your examination purpose but for also for your knowledge in the medicinal chemistry course. Okay, so I will first start with that summarization portion, and then I will proceed to the question answer session later. So the topics which I will try to summarize today, it will be basically mainly from the all the therapeutic agents like your antibacterial, anti-cancer, antiviral, antiviral, anti-cancer, and also the anti-hypertensive drugs and so on. So I will discuss the topics from all that particular courses and also I will discuss a little bit about various cellular mechanisms, especially mainly from your protein synthesis, the translation process, and also from the DNA replication and the different enzymes that are involved in this course. The reason of choosing these topics is like the therapeutic agents topics is very important. The antiviral, antifungus, anti-cancer drugs, these are all very important topics of medicinal chemistry. But before going directly to that topics, what the things you need to know is that various ways of the translation and transcription and all the your DNA synthesis, replication, and the enzymes involved in it, because most of the your antibiotic agents, let's say, or antiviral or any kind of your anti-cancer agents, mostly they use the cellular mechanism of the organisms mainly to tackle this kind of diseases. Okay, so I thought it will be better if I can include that topics, because these were not very much discussed in your course, but I feel like this can be a very good topic for the medicinal chemistry also. So just let me check once more. Yeah, so again I hope my voice is clear, right? So if you have any problem regarding my voice or the presentation, let me know in the chat box, and during the session if you have any question, any doubt, you can also let me know here. Okay, at the end of the session, again today, since today it is our last session, okay, so there won't be any live interaction class from next week. So as today is the week 12 live session, but at the end I will share my email ID, so, and I think most of you have my email ID. So after the sessions also, if you have any problem, you can just mail me. Okay, I will try to help you. So there is no problem regarding that. And also if you get time, so after today's class, try to give a feedback in the SWAM portal. I will show you where to give the feedback, so it will be helpful for me to improve from the next semester. So I will do it according your feedbacks also. So let me start with the topics today. So the first topic which I will discuss today is the process of protein synthesis. So now we all know that the protein synthesis, how the protein synthesis happen from the DNA. We have a template from the RNA template, and that RNA is responsible for protein synthesis, and the process of protein synthesis is known as the translation process, right? So DNA in nucleus as a template, it acts as a template. There is a mRNA which is processed and released into the cytoplasm. From this mRNA binds to the ribosomes. tRNA that carries amino acid to mRNA, anticodon codon complementary base pairing occurs. Peptide chain is transferred from resident tRNA to incoming tRNA. tRNA differs and protein modification happens after the translation process. So basically the process of protein. So basically RNAs are responsible, mRNA, tRNA, these are mainly responsible for our protein synthesis. So how it happens basically, so here as you can see, there is a process of protein synthesis. So there are three parts of the translation process. First one is the initiation, then we have the elongation, and then we have the termination, right? So what happens, so this is your mRNA template. This mRNA template is generated from DNA. So this goes from 5 prime to 3 prime. At the 5 prime end we have this start codon, which is AUG codon. What are codons? I have already discussed you that it is a 3 base pair your sequence which is known as codon, and this 3 base pair sequence which is a codon, it translates for a particular amino acids. There are several codons that different codons can translate for a same amino acid. Okay, so it can happen. So there is a particular codon which is called the start codon, because always the protein synthesis starts with this codon. So whenever there is this codon, it is like signal for starting the protein synthesis. So with this AUG, it codes for a particular amino acid. So which amino acid is coded by AUG or the start codon? Let me know in the chat box. And also we have 3 stop codons, right? UAG is given here. What are the other 2 stop codons? For which amino acid? AUG or methionine it codes for, and also let me know in the chat box. This first stop codon, one stop codon is the UAG. What are the other 2 stop codons? We have total 3 stop codons. since it is our last class, whenever I am teaching you, I will just ask you a few little questions. So I think it will be better that if you can answer, it will just you have a revise for the topics. Yeah, methionine is the first start codon. It is not a start codon. AUG is the start codon, and methionine is the amino acid for which it codes for. And what are the other two stop codons apart from UAG? UAA, very good. And what is the last one? We have three stop codons, right? UAA, UAG, and what is the third one, third stop codon? What is it? UAC. UAG I have shown you. UAC is the correct one. Think. UAA is the correct one. Then I have shown you UAG. So what is the other one? So the third stop codon is UGA. Okay, UGA is the third stop codon. So we have three stop codons, UAA, UAG and UGA. So these codon are the signal for the protein synthesis of growing protein chain to stop now. So whenever this kind of stop codons appear, the chain is terminated. Okay. So now how this process happens? So I have told you that there are three stages. First one is the initiation stage, then we have a elongation stage followed by a termination stage. So what happened during the first, we have this a tRNA with first amino acid, which is methionine, okay, and it has this tRNA has an anticodon sequence. Anticodon sequence is the complementary base pairs of the codon system, right? So and there is a large, so ribosome is the particular component of the cell component which helps during the protein synthesis. So the ribosome have two subunits, one large subunit and one small subunit. So can you tell me for prokaryotes what are the large and small subunits, and for the eukaryotes what are the large and small subunits for ribosome? These things I have already teached you in the class, okay. So I just trying to figure out if you remember these things or not. So I just asking you that for it. So what are the subunits of the particular ribosome? So one is large subunit and one is a small subunit, but there is a particular code you can say for the subunits. For prokaryotes is something and for eukaryotes is the different one. Read these things before examinations because these topics are important. So what are the subunits? Yes, very good. 50S and 30S is it's for the prokaryotes, right? And 60S and 40S it is for the eukaryotes. Very good. Yeah. See, that is good. I am just trying to making you remember the topics you have already read. So yeah, 50S, 30S and 60S and 40S for eukaryotes and 50 and 30 for the prokaryotes, okay. So these are those ribosomal subunits here we have. So first it is the initiation stage. So what happens during here? A translational complex form and the tRNA brings the first amino acid in polypeptide chain to bind to start the codon for mRNA, okay. Then we have the elongation stage. This stage goes longer. The tRNAs bring amino acids one by one to add to the polypeptide chain like this, okay. There is amino acids, this circular ones, and this is the tRNA. One by one the tRNA, they come, they add the amino acids to particular chain, growing chain, and then they leave. And then at last we have a particular termination step. So what happened here? Release factor recognizes the stop codon, translation and complex dissociated and completed polypeptide chain is released. So what happens during this? Whenever there is a stop codon appears, there is a release factor is known. To this release factor basically recognizes the stop codon and it gives a signal for stopping the polypeptide chain, and synthesis and growing chain is released and the complete translational complex is first dissociated, and later on when there is another protein synthesis happens, another translational complex it forms and then another polypeptide chain formation happen. So when the circle is completed, the components are recycled for a new protein cycle. So this is the translational cycle or the process of protein synthesis. The reason of me to showing you this, so if you remember there are few antibiotics or several antibiotics which targets the protein synthesis, the bacterial protein synthesis, to stop the growth of the bacteria, right. So that's how you should know how the translation process happens, so you can more clearly understand how that particular thing is stopped by the antibacterial agents. So what is the difference between transcription and translation? So transcription is the process of transcribing a piece of DNA into RNA, and the enzyme involved in transcription process is the RNA polymerase. Okay, from DNA to RNA, the process is known as the transcription, versus the translation. It is the process that involves the synthesis of a protein from a mRNA template, converting the mRNA code into an amino acid sequence within a protein. So this is known as the translation process. So there is the difference between transcription and translation. Don't get confused between these two. Okay. Now I will move toward the next slide. So this was the translation process or protein synthesis process. Now another very important process is which happens during our in our body is the DNA replication process. So how the DNA replication process happens, it was not directly written in your course, okay, but there were a little bit about the different enzymes which are involved in the DNA replication. So I thought to give you an overview. So how the DNA replication happened. So this is the parental DNA. This in this portion, this is the parental DNA. From this, the two different DNA strands are formed. Okay. So the DNA strands, one goes to 5 prime to 3 prime and another goes to 3 prime to 5 prime, and the replication which happens always toward the 5 prime to your 3 prime portion. Okay. The 5 prime to 3 prime portion, the replication happens. So as you can see, this is known as the your DNA. So there are several enzyme, and there are two different strands. So as you can see, there is one strand is here and another strand is here. This dark blue color one is the parental DNA, and the light blue color one is the your particular new DNA which is DNA chain which is forming, right. So there are two strands. One is known as the leading strand, which is synthesized continuously from 5 prime to 3 prime, and one is lagging strand. With the lagging strand is also synthesized from 5 prime to 3 prime, but the synthesis is not a continuous form, it is a discontinuous synthesis. So how it happens, let us start from here. So first there is an enzyme which is known as helicase. What does this helicase enzyme do? This helicase enzyme unwind the parental double helix DNA. So this is the double helix DNA to start the replication. So what it has to be done, like the particular there should be the base pair, hydrogen bonding between the two base pair of two different DNA strand, it should be broken. Okay, and then it has to, and so that those single strands can be formed. So helicase is the enzyme which takes part in this work, and it unwinds the parental double strand DNA to form the single strand. Okay, and DNA topoisomerases are the enzyme. What does the DNA topoisomerases do? They basically relaxes the particular your tension which it generates from here during the unwinding. Then we have small single strand binding proteins. These proteins stabilize the unwound parental DNA. These are the your single strand binding proteins. Then we have this strand which goes from 5 prime to 3 prime. So this is your leading strand. Okay, so the leading strand is from 5 prime to 3 prime continuously. Okay, then the enzyme which helps for synthesizing this particular leading strand is only the DNA polymerase. But there is another strand which is the lagging strand which is synthesized discontinuously. The primase synthesizes short RNA primer. So there is an enzyme which is known as the primase. The primase synthesizes, this is the primase enzyme, it synthesizes this red one is known as the RNA primer, which extended by DNA polymerase to form the Okazaki fragment. Okay, so these are the Okazaki fragment. So now after RNA primer is replaced by DNA by other RNA polymerase, so the DNA ligase joins the Okazaki fragment to a growing strand. So the DNA ligase is another enzyme. What does it do? The first RNA primer, it forms short DNA of fragments which are known as the Okazaki fragments, and these fragments are continuously formed to a single strand, and this formation also goes from 5 prime to 3 prime, but the formation is discontinuous in the lagging strand. Okay. And after the RNA primer is replaced by DNA, there is DNA ligase enzyme which is this one. It joins the Okazaki fragments to form another particular your DNA strand. And overall direction of replication is, as I have already mentioned, the overall direction is like this one. Okay, like this 5 prime to 3 prime. The overall direction is 5 prime to 3 prime. The 5 prime to 3 prime in leading strand, remember it is the continuous, the lagging strand is discontinuous, and also the enzymes. So in the DNA replication, basically the important part is the different enzymes, the role of different enzymes. So already I have mentioned you various enzymes which takes part in DNA replication. We have helicase, we have primase here, we have DNA polymerases here, we have ligase here, and also we have DNA topoisomerase and so on. So I will now I will just summarize the different enzymes which are involved in DNA replication, and also their work and how they do it. So DNA ligase, I have mentioned, in the DNA ligase seals the nicks between the Okazaki fragments, converting them to a continuous strand of DNA. Covalently closes the nicks of double stranded DNA. The DNA ligase also closes the nicks in double stranded DNA which are formed. Then we have helicase. Helicase uses energy from ATP to break the hydrogen bonds holding the base pairs together. This allows two parental strands of DNA to begin unwinding and forms two replication forks. Each strand of parental DNA has its own helicase. In humans, two inherited diseases, Werner's syndrome and Bloom syndrome, result from the helicase defects. E. coli contains at least 6 different helicases, some involved in the DNA repair and others in the conjugation. The principal helicase in DNA replication is DNA B. Then we have DNA polymerase. DNA polymerases are enzymes that synthesize DNA molecules from deoxyribonucleotides, the building blocks of DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from a single original DNA molecule. During this process, DNA polymerase reads the existing DNA strands to create two new strands that match the existing ones, also performs proofreading and error correction. We have primase. DNA primase is an enzyme involved in the replication of DNA and is a type of RNA polymerase. Primase catalyzes the synthesis of short RNA or DNA in some organisms, segment called a primer complementary to single strand DNA template. Primase is of key importance of DNA replication because no known replicative DNA polymerase can initiate the synthesis of DNA strand without the initial RNA or DNA primer for temporary DNA elongation. After the elongation, the RNA piece is removed by 5 prime to 3 prime exonuclease and refilled with a DNA. And topoisomerase, every cell has enzyme that increase or decrease the extent of DNA unwinding is known as the topoisomerases. Topoisomerase is also known as DNA guidase and they act on the topology of DNA. Topoisomerases bind to the double stranded DNA and cut the phosphate backbone of either one or both DNA strand. This intermediate break allows the DNA to be untangled and unwound, and the end of the process is the DNA backbone resealed again. Topoisomerases is an enzyme that can change the linking number. Okay. So these are all the enzymes. Okay, these are all the enzymes that are important, very important in case of your DNA replication, and also the process itself is a very important one, the DNA replication process, which I have mentioned already. So till now, if you have any problem, any doubt, you can let me know. If I need to repeat one any topic till now which I have told you, you can let me know. So I think it is clear how the translation process happens and how the DNA replication process happens, or do I need to repeat anything? Okay, so I think it is clear. Then I will move forward the next topics which I want to summarize today. Okay, let us move forward. So now what I will discuss is, so this I have already mentioned you. So now antibiotics, we all know antibiotics or the antibacterial agents that are the very important class of therapeutic agents which we have already discussed. So there are different classes of the antibiotics, right? We have discussed several classes also, all the classes of antibiotics, the classifications, the method of action and the examples. Okay, these are very important, and also few structures if you can remember. So this is very important. The structures I am telling you to remember, because sometimes the questions can be given like with only the structure. Let's say you are given a particular structure and you are asked to write the mechanism of action of that particular antibiotic agent. So if you remember that structure in which class of the particular antibiotic it is into, then you can easily answer that question. Okay, you don't need to remember all the particular the exactly structure, but the main top points of the structures. Like for example, this the first, so let us first discuss about different antibiotics. Okay, so the different classes are, for the most important and the well known is the penicillins, then we have cephalosporins, we have quinolones, we have macrolides, and we have tetracyclines. Okay, so this penicillins are a very important class of, so there are different generation. So the first generation penicillins are penicillin G, followed by amoxicillin, then we have triclosan and piperacillin. Okay, then for cephalosporin, the first generation is cephalotin, then followed by cefuroximine, then we have ceftazidimine and cefepime. Then for quinolone, the most important and the simplest one is the lanulixic acid, followed by ciprofloxacin, levofloxacin, and moxifloxacin. Then for macrolide, erythromycin is the well known antibiotic agent, right? Then we have the clarithromycin and telithromycin. And then we have the particular tetracyclines like oxytetracycline, doxycycline, and tigecycline. So the structures for that I have told you. So if you can look at the structure carefully, this portion which is drawn in black, it is the common for every antibiotic, right? So this is for penicillin, we have a beta lactam ring, then it is fused with another 5-member ring which have a sulfur, and this is the overall, this is the main component of the penicillin antibiotic. For cephalosporin, it is also a beta lactam antibiotic which has a beta lactam ring, but here the ring which is conjugated with this beta lactam ring is a 6-member ring sulfur containing. So if you are given this structure, if you look at the carefully, if you remember, like yes, cephalosporins have this kind of structure, then you can easily answer that particular question, right? Then we have quinolone, we have a quinoline ring. Okay, and this quinoline ring which have a 4 position, it has a ketone group. Okay, that's why it is known as quinolone. Then if you are given this kind of structure, if you identify, yeah, this is a quinolone, so also the quinolone has a as 1 to this 3 position, C3 position, you have a carboxylic acid. So if you are given any kind of this quinolone ring with the C position, it has a carboxylic acid, then you know it is a quinolone class of antibiotic. And if you remember what is the method of action of that particular quinolone, you can answer the question carefully or easily. Then we have the macrolides. Macrolide means actually macrolide mean it has a large ring. Okay, a very large ring like this. This is the basic structure of macrolide, which has a 1 2 3 4 5 6 7 1 2 3 4 5 6 7 8 9 10 11 12 13 C13. Okay, 13 carbon chain, and also oxygen is here. So it is like this macrolide, and also it has a sugar moiety here, right? So if you are given this kind of structure, you can understand, yeah, this is has a the C13 or C14 14-member ring with some sugar unit, so it must be a macrolide kind of. And tetracyclines, the name itself suggests it has a 4 ring for 4 cycles which are attached with each other. It is known as tetracyclines. Okay, so these are the main and important class of antibiotics. Now whenever we are reading about the antibiotics or antibacterial agents, the topics which we need to focus on is the method of action or mechanism of action of that particular class. So the mechanism of action, so as I have that's why I have mentioned the translation and also your transcription process and the DNA replication process earlier, because these are the translation process, the DNA replication process, the different enzymes that are targeted in particular mechanism of action of different antibiotics. So the first and the foremost and very important bacterial you can see a mechanism which is targeted by the antibiotics is the cell wall synthesis. The bacterial cell wall synthesis, it is disrupted by various important antibiotics like cycloserine, vancomycin, bactricin, penicillin, cephalosporin, monobactam, carbapenems. These are the antibiotic agents which the mechanism of action is the inhibition of the cell wall synthesis, right? Then we have the inhibition of folic acid mechanism. I have already discussed it in the last class itself, I think. The folic acid mechanism, it can be, so we in the case of folic acid metabolism, what it has a PABA is the starting point, which is the para-amino benzoic acid, from which the DHF and THFs are formed, okay, and the from PABA to DHF, this step is inhibited by the sulfonamides, and the DHF to 2HF, this particular portion or this particular step is inhibited by trimethoprim. And this is another very good example of which kind of example of enzyme inhibition. Last class itself I have discussed it, okay, which or last to last class I have discussed it itself with this example, the trimethoprim and sulfonamide. Which type of inhibition is this pathway? Does anybody remember? Which kind of inhibition is this particular where the trimethoprim and sulfonamides work together to inhibit the folic acid mechanism? You can let me know in the chat box. If you remember, you can let me know, or if it is wrong, it is fine, but I am just figuring out that if you have look at, yeah, sequential blocking, very good. Sequential blocking is the process, okay, where the two different molecules or two different drugs or inhibitors are used to inhibit a same pathway, okay, to have a higher effect. This is known as a sequential blocking, very good. So, these are the trimethoprim and sulfonamides, these antibacterial agents, they inhibit the folic acid mechanism of bacteria. Then we have the cytoplasmic membrane structure and function, which can be disrupted by the polymyxin and daptomycin. Then this is the cytoplasmic membrane, this is the cell wall, this is the plant. So, lipid biosynthesis is inhibited by plantanomycin, okay. Then the protein synthesis, by the tRNA, this is inhibited by the mupirocin and puromycin, okay. As you can see, here is the 50 and 30S ribosome of the prokaryotes, which are the bacteria are prokaryote. The protein synthesis, the 30S inhibitors are the tetracyclines, pectiomycin, streptomycin, generamycin, kanamycin, amikacin and nitrofurans. And we have the protein synthesis of 50S inhibitors are the erythromycin, chloramphenicol, clindamycin and lincomycin, okay. These are the 50S inhibitors. Then DNA directed RNA polymerase inhibitors are rifampicin and streptoviricins. Then we have the RNA elongation, which is inhibited by actinomycin, and DNA guidase inhibited by the quinolones like nalidixic acid, ciprofloxacin, novobiocin. So, these all are the very important of the important pathways or mechanism of actions of different antibiotics. So, this is the very important. So, remember what is the classes of antibiotic agents, what are the mechanism of action for a particular class, and also few examples of that particular class, and also the structural features which are important. So, I will suggest to all of you to read these things very carefully and try to remember, okay. So, now I will move toward the next topic. Antifungal agents were not very much included in your topic, but I thought because you are reading about antibacterial, antihypertensive and anti-cancer and all this type of drugs, so you should also know about little bit about the antifungal drugs. So, antifungal drugs are the medicine that deployed in the treatment of mycose. Fungal infection of the skins are, as of 2010, is the 4th most common disease in the world, affecting up to 1 billion people. So, different antibacterial agents are available. So, the first is the imidazole class. So, imidazoles are the nitrogen containing heterocyclics, right. So, nitrogen containing heterocyclics, imidazoles, these are the main pharmacophore of this kind of antifungal agents. So, imidazole have a 5-member ring where the nitrogens are 1 and 3 position. So, the imidazole containing antifungal groups are the clotrimazole, oxyconazole, miconazole, iconazole, ticonazole and ketoconazole. Then we have triazoles. Triazoles are 5-member nitrogen containing heterocycle which have 3 nitrogens, okay. These are the examples are fluconazole, itraconazole, tetraconazole, voriconazole, isavuconazole and posaconazole. These are the azole antifungals which inhibit the lanosterol 114 alpha demethylase, the enzyme required to convert lanosterol to ergosterol, okay. And this is the very important class of antifungal, your azole antifungals, okay. Then we have the griseofulvin, which inhibits the mitosis in the dermatophytes. It is ineffective when applied topically. We have echinocandins, or which is the caspofungin, anidulafungin, and bicafungin. So these inhibit the cell wall synthesis by targeting glucans 1,3 beta glucan synthesis. Then we have flucytosine, pyrimidine analog converted into 5-fluorouracil by the fungal enzyme cytosine deaminase, active against the yeast infection. Then we have pauleins, which are the natamycin, amphotericin B, or nystacin. Okay, and we have allylamines, which are the amorolfine, natifine, and terbinafine. They inhibit the squalene epoxidase. So these are different antifungal agents and their mechanism of action. So mycosis is a fungal infection in animal and including human. Mycosis are the very common and variety of environmental pathological condition can contribute to development of fungal diseases. Okay. Then triazoles are very important and newer but less toxic and more effective class of antibiotics. Okay, we have fluconazole, itraconazole, albuconazole, posaconazole, and voriconazole. Like this, we have, this is the structure of fluconazole. So I have mentioned these are the triazole ring. This is the 1 to 4 triazole. So we have two kind of triazole. One is 1 to 4, 1, 2, and 4 that are based on the position of nitrogen, and one also can be now 1, 2, 3 where the nitrogens are side by side. Okay, so like other imidazole and triazole class of antifungals, fluconazole inhibits the fungal cytochrome P450 enzyme 14 alpha demethylase. Mammalian demethylase activity is less sensitive to fluconazole than fungal demethylase. This inhibition prevents the conversion of lanosterol to ergosterol, an essential component of the fungal cytoplasmic membrane, and subsequent accumulation of 14 alpha methyl sterols. Fluconazole is an inhibitor of human cytochrome P450 system, particularly the isosome CYP2C19, CYP3A4 and CYP2C9 to lesser extent. In the theory, therefore, fluconazole decreases the metabolism and increases the concentration of any drug metabolized by these enzymes. In addition, its potential effect on QT interval increases the risk of cardiac arrhythmia if used concurrently with other drugs to prolong QT interval. Okay. This was an overview about a fluconazole, which is a very important class of triazoles. So now, okay, one second. So now we will move toward the next topic. So we have different kind of antihypertensive drug. Antihypertensive drugs, it works on the blood pressure regulation of our body, right? So if somebody has a higher blood pressure, and also it helps in different kind of heart conditions also. So so the classes of antihypertensive drugs along with the examples and the mechanism of actions are very important. In any kind of your therapeutic agents, be it antibacterial, antifungal, antihypertensive, or any kind of this kind of particular topics, the mechanism of action, example, and classifications are very important. Each and every class you have to remember few examples and also the mechanism of action and a little bit of structure, and it is same for each and every classes. Okay. So we have the diuretics at first, which are the very common and initially used antihypertensive agents like thiazides, hydrochlorothiazide, and chlorthalidol, high ceiling, the fluorosemide, K plus aspirin, the spironolactone, tramterine, and amiloride. Amiloride is a very common and very well known diuretics. The mechanism of action is it acts on kidneys to increase the excretion of sodium and water, decrease in blood volume, and decreased blood pressure. Angiotensin converting enzyme inhibitors or ACE inhibitors, the example captopril, lisinopril, enalapril, ramipril, and fosinopril. So these are the angiotensin converting enzyme or ACE inhibitors. The mechanism of action is inhibit the synthesis of angiotensin 2, decrease the peripheral resistance on blood volume. Angiotensin and AT1 blocker, the losartan, candenosertan, valsartan, telmisartan, these are the angiotensin angiotensin AT1 blockers that blocks the binding of angiotensin 2 to its receptor. And we have adrenergic receptor antagonists, or known as the beta blockers, like propranolol, nebivolol, atenolol. They competitive antagonists that block the receptor side on androgenic beta receptors, and beta blockers are also very good for your regulating the blood pressure along with the heartbeat. Okay, the beta blockers are very important class of antibiotics, and which acts on this dual kind of things. So now let me check if everything is fine. So if you have any problem till now, which whatever we have discussed, let me know. Otherwise, I will continue. So till now, if you have any doubt or any problem or any question, or you want me to repeat anything, you can let me know, okay? Or everything is fine, then I will proceed to the next topic. Okay, so I think I can proceed to the next topic, right? So next we have the antiviral drugs or antiviral agents. So HIV-1 is the most of, in the case of antiviral drugs, so there are different antiviral agents. So but today I will discuss mainly regarding the HIV, okay, because HIV is the most common type of HIV is the most common antiviral and very important antiviral disease, okay. So all of you know HIV, till now it doesn't have any particular your like vaccination or something kind of thing. So that's why it is a very crucial for us to learn about the HIV. So HIV-1 is the most common type of human immunodeficiency virus. It attacks your body's immune system. The virus destroys the CD4 cells. These cells help your body to fight infections. HIV-1 can severely damage your immune system and lead to acute immune deficiency syndrome, which is the AIDS. CD4 cells are white blood cells that play an important role in immune system. The CD4 cell count gives you an indication of health of your immune system, your body's natural defense system against the pathogen, infection and illness. Currently there is no cure for HIV and AIDS, as I have told you. Once you have the infection, your body cannot get rid of it, okay. However, there are many medications that can control HIV and prevent the complication, and these medications are known as antiretroviral therapy, okay. So let us see what are the different kind of therapeutic agents which can help in tackling the particular HIV. So reverse transcriptase inhibitors. So the reverse transcriptase is an RNA dependent DNA polymerase that utilizes a strand of RNA to synthesize double stranded viral DNA. This mechanism helps the retroviral gene to get incorporated into the host cell's gene, and various RT inhibitors are developed which, on the basis of the binding site and chemical class of compounds, are classified into NRTIs, which are the nucleoside reverse transcriptase inhibitor, and NNRTI, which are the non-nucleoside reverse transcriptase inhibitors, okay. So what are the NNRTIs? So non-nucleoside reverse transcriptase inhibitors. Among the HIV RT inhibitors, which are the HIV-1 reverse transcriptase inhibitor, non-nucleoside reverse transcriptase inhibitor or NNRTIs have gained a definite place in due to their unique antiviral potency, high specificity, low toxicity in antiretroviral combination therapies used to treat HIV. We have NRTIs, which are the nucleoside reverse transcriptase inhibitors. While NRTIs and NNRTIs are alike and effective at terminating DNA synthesis and HIV replication, NRTIs or the nucleoside reverse transcriptase inhibitors inhibit the reverse transcription by causing the chain termination after they have been incorporated into the viral DNA. This was the first group of antiretroviral agents to be used against the HIV. NRTIs lack a 3' hydroxyl group of the dioxyribose moiety, as a result, following incorporation of an NRTI, the next incoming deoxynucleotide cannot form a 5' to 3' phosphodiester bond that is needed for a extend to DNA chain. Thus, where NRTI is incorporated, viral DNA synthesis is halted, is process known as a chain termination. All NRTIs are classified as a competitive substrate inhibitor. This is very important, okay. NRTIs are classified as competitive substrate inhibitor. Then we have the protease inhibitor. Protease inhibitor is a very important class of HIV for tackling the HIV disease. The protease are the enzyme that cleave the protein molecules into smaller fragments. HIV protease is vital for both viral replication within the cell and the release of mature viral particles from an infected cell. Various protease inhibitors are developed for HIV treatment. For some of the FDA approved drugs of the classes, saquanavir, indinavir, atomazavir, ritonavir, bit, and so on. Okay, so whenever you are reading about different inhibitors, like I haven't given any example, forgot to give the examples of NNRTIs and NRTIs. So just remembering the mechanism of action and also the particular examples are very important, because question can be directly given to from the example also, that the particular this is a particular for example, you can, the question can be given like indinavir is a which kind of HIV agent. Okay, is it a protease inhibitor, NRTIs, NNRTIs, and so on. Okay, all write the mechanism of action of this the name of the drug. As the question, this questions can be given like this. So remembering what is the particular inhibitor, what is the mechanism of action, and again what is the example. For antiviral agents, the reverse transcriptase inhibitor and protease, so these structures are very complicated, so you do not need to remember the structures of the same. Okay, but remember what are the example and what is the mechanism of action. Now I will discuss about the anti-cancer drugs. So anti-cancer agents are a very important topic in our medicinal chemistry. Okay, the anti-cancer agents are basically the right now the most of the research are, I mean the very extensive part of research in medicinal chemistry, it happens regarding your anti-cancer drugs or the anti-cancer for development of anti-cancer drugs. Okay, so because sometimes what happens, like the mutation rate is very high right now, so the different the anti-cancer drug after some time it gets the body gets immune and the cancer cell get immune against that particular drug. Okay, so anti-cancer agents or anti-cancer drug targets keep changing from time to time to have the more potent drugs. So what are the key points? So today I will discuss different key points of the I mean the for anti-cancer targets, anti-cancer drug, and different anti-cancer drug targets. So the key points, like cancer cells have defects in the normal regulatory controls governing the cell growth and division. Such defects arise from the mutation regarding in the activation of oncogenes and the inactivation of tumor suppression genes. Defects in signaling pathway are commonly found in cancer cells. The pathway stimulating cell growth and division are overactive as a result of overproduction of a crucial protein in the pathway or the production of abnormal protein. The proteins involved include the growth factors, receptors, signal proteins, and kinases. The production of regulatory proteins which suppress cell growth and division are suppressed in many cancers. The cell cycle consists 4 phases. We have already discussed the 4 phases of cell cycle. Like what are the 4 phases of cell cycle? Does anybody remember? I have discussed it in the I mean the week I think it was in your week 11. So I think I have discussed it in our 10th live session. So what are the 4 phases or the 4 stages of the your cell cycle? Will antifungal drugs, so what is the question? I didn't get it. So can you repeat your question? Antifungal drugs was not included in your syllabus directly, so I don't think there will be many questions regarding antifungal drugs. I have just given you so that you just learn these topics, okay, or learn these things. But it is not directly involved in your course, so the broad question won't be come, or the subjective question won't be come from the antifungal agents. Okay. So does anybody remember what are the 4 cycles of cell cycle? What are the 4 stages of cell cycle? I have discussed it already. The cell cycle is when the cell prepares to division, for cell division, right? So what are the 4 stages? Let me know in the chat box if you remember it. Yes, G1, S, G2 and M. Very good. So these are the 4 stages of the cell cycle. So the cell cycle, okay, 1 second. So the cell cycle consists of 4 phases. Progression through the cell cycle is controlled by cyclins and cyclin dependent kinases, moderated by restraining proteins. Defects in this system can be detected in 90% of cancers. Apoptosis is a destructive process leading to cell death. Cells have monitoring systems which check the general health of the cell and trigger the process of apoptosis if there is too many defects. Regulatory proteins have a moderating influence on apoptosis. Defects in apoptosis increase the chances of defective cells developing into cancerous cells and reduce the effectiveness of the several drugs. Telomeres act as a splices to stabilize the end of the DNA. Normally, they decrease the size at each replication until they are too short to be effective, resulting cell death. Cancer cells are active in expression of an enzyme called the telomerase to maintain the telomere and become immortal. Angiogenesis is a process by which tumors stimulate the growth of the new blood cells to provide the nutrients required for continued growth. Agents which inhibit the angiogenesis are useful in anti-cancer therapy to inhibit the tumor growth and to enhance the effectiveness of the other drugs. Metastasis is the process in which the cancer cells break free of the primary tumor, enter the blood supply and set up the secondary tumors for other tissue. To do this, regulatory controls which fixes the cell to a specific environment and which destroys cells become detached and overruled. Surgery, radiotherapy, chemotherapy are used to treat cancer. Chemotherapy usually involves combinations of drugs having different targets of mechanisms or action. Traditional anti-cancer drugs are generally cytotoxic and our modern drugs are selective in action. Cancer cells can have intrinsic and acquired resistance to anti-cancer drugs. Resistance may due to poor uptake of the drug, increased production of the target protein, mutations which provide the drug binding to its target, alternative metabolic pathway or efflux systems which expel the drugs from the cell. Intercalating drugs contain planar, aromatic or heteroaromatic ring system which can slide between the base pairs of DNA or double helix. Alkylating agents continue electrophilic groups which react to the nucleophilic centers of DNA. Two electrophilic groups are present, interstrand or the intrastrand cross-linking of the DNA is possible. Nitrogen musters react with guanine groups of DNA to produce cross-linking. The reactivity of the agents can be lowered by attaching electron withdrawing groups to the nitrogen to increase the selectivity against the DNA over proteins. Incorporation of important biosynthetic building blocks aid the uptake of the rapidly divided cells. Cisplatin and its analog metallitic agents which cause the intrastrand cross-linking, they are commonly used for treatment of testicular and ovarian cancers. CC1064-65 analogs are highly potent alkylating agents which are being considered for use of antibody drug conjugates. Calicimycin is a natural product which reacts with the nucleophiles to produce diradical species. Reaction with DNA ultimately leads to cutting the DNA chain. Antisense molecule have been designed to inhibit mRNA molecule to coat the proteins which suppresses the apoptosis. So, all these things, the cisplatin, the intercalating agent, alkylating agents, okay, calicimycin, all I have discussed already. If you look at the PPTs earlier which I have shared, you will find all the examples I have given, okay, in our question answer sessions. So, I thought to just give you an overview of the anti-cancer drug and different drug targets and also little bit about the cancer cells. Okay, then we also have this this also are in your anti-cancer agent. Antimetabolites are the agent which inhibit the enzymes involved in the synthesis of DNA or building blocks. Thymidylate synthesis catalyzes the synthesis of DTMT from DUMP. The cofactor required for this reaction is regenerated by the enzyme dihydrofolate reductase. Inhibition of either enzyme is useful in anti-cancer therapy. Ribonucleotide reductase catalyzes the conversion of ribonucleotide diphosphate to deoxyribonucleotide diphosphates. It can be inhibited directly by drugs and directly by drugs or indirectly by inhibiting adenosine diamminose. In later case, the build up the ADP results in the allosteric inhibition. Various nucleosides and purines act as a prodrugs and are converted in the cell to agents to inhibit the DNA polymerases. The active agents also acts as a substrate and incorporated into growing DNA, leading to chain termination or the inhibition of the replication. Hormone based therapies are used against cancers which are hormone dependent. Hormones can be administered which counteract the offending hormone. Alternatively, anti-hormonal therapy compounds are administered to prevent the offending hormone from binding to the receptor. Glucocorticoids, estrogens, progesterones, androgens, LH, RH are used in hormone based therapy. Agents which act on receptor antagonists are used to block estrogens and androgens. Enzymes inhibitors are used to block synthesis of hormones. An important target enzyme aromatase which catalyzes the last step of leading to estrogens. Then we have agents which inhibit the polymerization or depolymerization of microtubules are the important class of anti-cancer agent like vinca alkaloid, podophyllotoxin, combristatins, and a variety of natural products bind to tubulin and inhibit the polymerization process. Plexitaxel and its derivative bind to tubulin and accelerate the polymerization by stabilizing resulting microtubules. Newer analogues are being investigated which show better oral bioavailability, improved pharmacological properties, and activity against drug resistant cancers. A variety of natural products have been discovered which have a similar mechanism of action of to plexitaxel. Then we have matrix metalloproteaminase or MMPs, which are zinc dependent enzyme which degrade the extracellular matrix and encourage the process of angiotensin, tumor progression, and metastasis. Inhibitors of MMPs have been based on the small peptide sequences and protein substrate. In general, the susceptible peptide bond have been replaced by a stable bond. Subsequent have been incorporated to fit binding substrate and ligand to zinc cofactor. Second generation inhibitor of MMPs are non-peptide in nature and more selective in action. The proteasome is a destructive enzyme complex that breaks down proteins. Inhibition leads to the buildup of completely regulatory protein which triggers apoptosis. Histone acylases and deacylases are involved in regulation of transcription. Inhibitor of histone deacylase are in clinical trials. A variety of other enzymes are potential target of novel anti-cancer agents. A large number of synthetic structures have natural products have anti-cancer properties of unknown mechanism. Others appear to work by working in different mechanism. Protein therapy has been proved to in treatment of various certain cancers. Okay. Then there are the various clinical aspect of antibody and antibody drug conjugates, like for example we have the trastuzumab which is a humanized monoclonal antibody which targets the extracellular region of HER2 growth factor and approved in 1998 for the standard first line treatment of HER2 positive metastasis breast cancer in combination to paclitaxel. HER2 is a member of EGFR family of tyrosine kinase receptors which overexpress in 25% of breast cancers. When the antibody binds to the receptor, it induces an immune response to attack the specified cell which promotes the internalization and degradation of the receptor, and this drug is given by injection and unfortunately it cannot cross the BBB or the blood brain barrier, ineffective against tumor cells that have been metastasis into the brain. Drug resistance and cardiac toxicity are the further problem in this case. Then we have the alemuzumab which is a immunized antibody that lyses the beta lymphocytes and for the B cell chronic lymphocytic leukemia where the therapies cannot win work. Antibody binds to the receptor CD552 antigen which found both on the normal and cancerous cells. Okay. Then we have the rituximab which is a chimeric antibody targeting CD20 receptor on beta lymphocyte, and it was approved in 1997 for treatment of diffuse beta cell and non-Hodgkin's lymphoma and follicular lymphoma. This causes lysine of beta lymphocyte. Patients should be monitored very closely as the reported fatalities the release of cytokines. In 2010 it was approved for FDA for the treatment of chronic lymphocytic leukemia. All these examples I have just given for your understanding, okay, because antibodies and antibody drug conjugates are very newer topics in case of anti-cancer drug discovery. So I thought to give you a little bit overview about this, and also I will discuss about kinase inhibitors which are comparatively newer anti-cancer drug targets. Okay. So all these things are for your knowledge. I don't think you need to memorize everything, but just read because these things are very important for understanding. Then we have the cetuximab which is a chimeric monoclonal antibody targets for extracellular domain of HR receptor, blocks the EFG from binding, used to be alongside with idanosine for treatment of metastasis collateral tumors which express EPGFR and which are proved resistant previous chemotherapy has been included irinotecan. The antibody is also alongside radiotherapy treatment of locally advanced cancer cell from head to neck. Then we have bevacizumab which are humanized monoclonal antibody that is given intravenously disable the growth of the VEGF required for angiogenesis. It is known to first line treatment of metastatic colorectal cancer along with the fluorouracil or folinic acid. Okay, it is also used in the first line treatment for the metastasis breast cancer along with the paclitaxel. We have ofatumumab which is target different epitope of CD20 receptor from rituximab and approved in October 2009. It is used to treat leukemia that cannot be controlled by other forms of chemotherapy. And we have panitumumab, which is fully humanized monoclonal antibody, targets epidermal growth factor and has been approved from treatment of colorectal cancer. Antibody drug conjugates, we have gemtumab, which have approved from treatment of acute myeloid leukemia and was withdrawn with 2010. We have this ibrutumumab, which was first approved for drug for radiotherapy, immunotherapy in treatment of non-Hodgkin's lymphoma. And we have then other this also there are different antibiotic drugs, which you can just read it later. So all these things I have taken from a book. Okay, so this is a very famous book of medicinal chemistry. The name I will let you know in the last. Okay, I can just share the name of medicinal chemistry. Apart from the lecture video and the presentation or the PPTs which I have given to you, you can just refer to a particular one important book, few important books of medicinal chemistry. Like we have a book, it is known as the Medicinal Chemistry, Introduction to Medicinal Chemistry by Graham L. Patrick. Okay, I will just give the name of the book here. You will find the PDF online. Okay, so you can just refer to these books apart from the presentation which I am giving, and also apart from the your lecture videos and the things. You can just refer to this book by So this is a very important book of medicinal chemistry. Somebody was asking me to give a last day on in the email that to suggest a very good book for medicinal chemistry. So I think Graham L. Patrick's book is very good. It is very preliminary but very understandable. Okay, so you can refer to this book for the medicinal chemistry. Then one more topic I will emphasize for the anti-cancer drug is the kinase inhibitors. I have discussed a little bit about it, but didn't get a chance to discuss about it very good. So protein kinase is an enzyme that catalyzes the transfer of phosphate groups from high energy phosphate donating molecules to specific substrate. This process is known as the phosphorylation, where the substrate gains a phosphate group and the high energy ATP molecule donates a phosphate group. The enzyme is involved in biochemical signaling pathways of the cell or effectors such as cell proliferation and apoptosis. Like substrate ATP, we have ADP and substrate with phosphate, and the enzyme which is responsible is the kinase enzyme. We have different kinase enzyme, like we have serine kinase, threonine and tyrosine. Why this particular three kinase? Because all these have a particular hydroxyl group. Okay, and this hydroxyl group is basically phosphorylated by the kinases. Like protein kinases are divided into mostly two parts. We have serine threonine kinases and tyrosine kinases. The serine threonine kinases, they have receptor serine threonine kinase like TGF beta, non-receptor serine threonine kinases like PK, PKC, MAP kinase, Aurora kinase. The tyrosine kinase, we have receptor tyrosine kinase like EGFR, PDGFR, FGFR, and non-receptor like BCR, ABL, CRC, etc. Human genome, we have 30 to 35k genes. For protein kinase gene 512 and protein phosphatase gene have 100. This is the reaction which is catalyzed by kinases. Then kinases, what does it do? Basically we have a protein which has a hydroxyl terminal hydroxyl group from serine, threonine or tyrosine. Protein kinases takes up a phosphate from ATP and forming the ADP, and we have this phosphorylated protein. This phosphorylated protein, what does it do? It provides some kind of signals which are these signals are responsible for different your cellular functions, like growth factor attached to the receptor. With the receptor cell division, tyrosine kinase start the signal and the cell divides like 2. Okay, and this kind of particular kinases are responsible for several cancer, and inhibiting the kinases can give rise to a very good range of anti-cancer drugs. So how this anti-cancer drugs work? So basically this is a hinge binding. So I think in the some class I have shown you the structure of an kinase enzyme, right? It has a N binding lobe, it has a C binding lobe, and the two lobes are joined by a particular strand of protein which is known as the hinge region. Okay, this hinge region is basically where the ATP binds. Okay, with and there is a kinase, the correct molecule to be kinase inhibitor. There are different type, like type A, type B. Okay, so the competitives are competitive kinase inhibitors are those which binds to the ATP binding or substrate binding pocket or ATP binding pocket of the particular kinase enzyme. We also have allosteric inhibitors or kinase inhibitors which bind not in the ATP binding pocket but to the other sides of the ATP binding pocket. So how the treatment of leukemia inhibition can be done by BCR-ABL kinase. So the inhibition, like this is the substrate this is a BCR-ABL. okay, and this is a substrate protein. So whenever there is a BCR-ABL kinase which catalyzes the transfer of phosphate group to substrate kinase, and it gives a signal for proliferation of survival of the cancerous cell, which gives the leukemia. But if the this is a glyphic is a very important BCR-ABL kinase inhibitor, which is a very famous anti-cancer drug. If glyphic binds to the BCR-ABL, what happens? The no cell signal happens and no leukemia happens. So this is for an example how the kinase inhibitors work. So now till I have discussed various topics of our medicinal chemistry, okay. I have discussed about your antibacterial, antifungal, and so other anti-cancer agents, and also the translation and replication process. So now I will show you few questions. Okay, these questions are very simple. I think all of you now can answer it, because we already have crossed our whole course, and we are at the almost the end of our course. So I think all of you can answer the questions. So today I will share few MCQs question. Like always, I will give you 4 options. You have to answer the questions in the chat box. I will check, and then I will let you know what is the correct answer, and we will discuss it, okay, little bit. So today it is the last class. I want all of you who are present in the class to respond and answer, okay. So let me show you the first question. So the first question of today's session is, which of the following statements true regarding the peptide bond formation? Number A, the carbonyl is seen to the hydrogen of NH group. Number B, carbon nitrogen bond is purely single bond in nature. Number C, ethoxyl is used to form peptide bond. And number D, hyperconjugation effect is reduced when hydrogen of NH bond is replaced by R groups. Which of the following statements is true regarding the peptide bond formation? I will give you 3 to 4 minutes, but all of you must answer the questions today, okay. Read the option very carefully and then let me know. Which of the following statements is true regarding the peptide bond formation? Let me check the chat box now. Okay, number B, number B, number B, number C, number A. So, okay, so I have different answers. Let me check which of the following will be the correct one. So, the statement which is true regarding the peptide bond formation. Number D, hyperconjugation effect is reduced when H of the NH group is replaced by R groups. So, this is a peptide bond, right? This is a peptide bond, the CO and NH group bond from the particular two amino acids are joined each other with the formation of the elimination of water, one carboxylic acid of one amino acids and amine group of another amino acid. So, let us look at the statements again. The carbonyl group is syn to hydrogen of NH. So, it is not true because the carbonyl group and the NH groups will be anti to each other, otherwise it will not get stabilized, right. The carbon nitrogen bond is purely single bond in nature. It is also incorrect because there is a double bond formation like this, a resonance structure where the lone pair of nitrogen is given here and this kind of positive negative charges happen. So, this is also untrue. Epmoxyl is used to form peptide bond. It is not true. There are different agents which help to form the peptide bonds like DCC, we have HBTU, HATU, so but not epmoxyl. But the last statement which is given like hyperconjugation effect is reduced when H of NH is replaced by I, it is true. Okay. Hyperconjugating effect means if let's look at this structure. So, this CO and NH are trans to each other. So, there is in this alpha positions we have the R groups, right. But if the R group this particular if the nitrogen is replaced, NH is replaced by R. So, the effect of the hyperconjugation which helps to stabilize the peptide bond, it will be reduced, okay, because R groups it will not show that particular hyperconjugation effect as of the hydrogen. So, that is why this last statement is the true, okay, the others are false. But all of you have answered, which is good, like most of you have tried to answer. So, now I will move to the next problem. Problem number 2 says what is the structure and ligand binding site of cholinergic receptor. Your options are number A alpha, beta, gamma, delta, epsilon and 2 beta. Number A, so this will be number B 2 into beta, alpha, gamma and delta and beta, gamma. Number C alpha, beta, gamma, delta, lambda and 2 gamma. And number D is 2 into alpha, beta, gamma, lambda, delta and 2 into alpha. What is the structure and ligand binding site of cholinergic receptor? These are for the structures and these are for the ligand binding site, okay, the subunits. Think and then try and let me know in the chat box. What is the structure and ligand binding site of cholinergic receptor? I will wait for 2 more minutes, then I will check and show you the answer. Let me check the chat box. Okay, number D, number D, number D, number C. So I have number B is A, number B, number C and number D. 3 different answer I have for this question. Okay, let me check. So what will be the structure and ligand binding site of cholinergic receptor? Number D, 2 alpha, beta, gamma, delta. It is the structure and the ligand binding site is 2 into alpha. So what is cholinergic receptors? So basically cholinergic receptors are present. There are different cholinergic receptor like M1, M2, M3. Then these kind of cholinergic like M1 is present in secretory glands like for salivation, stomach acid, swelling and lacrimation. M2 for heat, decreases the heart rate and bradycardia. M3 is for smooth muscle, GI, GU and respiratory contraction of smooth muscle like diarrhea, for bronchoemission and urination. M3 for pupil and ancillary muscle like contracts mitosis, increase the flow of aqueous humor. NM for skeletal muscle and plate, contraction of skeletal muscle. NN is the autonomic ganglia and adrenal medulla for secretion of epinephrine that controls ANS. So this is a schematic diagram which represents the receptor. Okay, this is the intracellular portion and it is the extracellular portion. So ligand binding sites are present in this 2 alpha position, right, and the structure is have alpha, beta, gamma, delta, 2 alpha, beta, gamma and delta. The effector domain in between, it is called the effector domain. Okay, then from this effector domain the different ions can pass through at the when their agonist is bind, the effector domain is activated and ions like sodium, potassium, calcium, chlorine, they can enter into the extracellular phase. Okay, so this is the structure and this is the ligand binding site. So, when two molecules of acetylcholines bind to the two alpha subunits, all the subunits move apart, opening a central pore of 0.7 nanometer, enough to allow passage of partially hydrated sodium ions. Anions are blocked from the passage, okay, through the channel by positive channel lining it. In other cases, K plus, C plus, the chrominous ion move through the channel depending on the ion selectivity. So, this is basically in case of mostly in case of your nicotinic cholinergic receptor. Okay, the molecule of 8 nanometer diameter is composed of 5 subunits that enclose a cylindrical cell tube, and in this particular nicotinic cholinergic receptor, this is only for your blocking passing for sodium, okay, and passage of anions are blocked, but depending on the other cholinergic receptor, the potassium, calcium, chlorines can also move through the channel. Okay, so remember the structure, what is the structure and ligand binding site of cholinergic receptor. So, now next problem. Problem number 3 says, which of the following statements regarding RNA is false? Your options are: number A, rRNA acts as a catalyst to make peptide bond. Number B, the amino acid sequence of proteins depend on the base sequence of mRNA. Number C, RNA polymerase 3 synthesizes mRNA. And number D, RNA polymerase 1 synthesizes rRNA. Which of the following statement regarding RNA is false? Read all the options carefully and then let me know in the chat box. Which of the following statements regarding your RNA is false? Which of the following statements regarding RNA is false? Let me check the chat box now. Number D, number D, number D. So, I mostly have number D as the answer, I think. Okay, let me check if number D is the correct answer or not. The correct answer will be number C. The statement regarding the RNA, which is the regarding RNA is false. The number C statement is false, which says that RNA polymerase synthesizes mRNA. So we have RNA polymerase. Let's discuss about RNA polymerase 1, 2 and RNA polymerase 3. So RNA polymerase 1, it polymerase that only transcripts for ribosomal RNA but not for 5S rRNA. It transcripts rRNA genes, okay, and resides in the nucleus and transcripts pre-rRNA at 20 copy to 100 copy. It contains same two non-identical alpha like subunit and insensitive to alpha amanitin. Then we have RNA polymerase 2, which is a central enzyme that catalyzes DNA directed mRNA synthesis during the transcription of protein coding genes. Transcripts for mRNA, miRNA, snRNA and snORNS genes, resides in the nucleoplasm, transcripts 25,000 genes, has 2 copies of a different alpha like subunit sensitive to 1 microgram per ml alpha amanitin. And then RNA polymerase 3, it transcribes small unscripted RNA, that is tRNA. It transcripts tRNA and 5S rRNA, resides in the nucleoplasm, transcripts 30 to 50 copies of gene contain 2 non-identical alpha like subunit and sensitive to 10 microgram per ml alpha amanitin. So the statement number C, which says that RNA polymerase 3 synthesizes mRNA, is false. RNA polymerase 3, it synthesizes or transcripts for tRNA genes, and for RNA polymerase 1 synthesizes the rRNA, and RNA polymerase 2, it synthesizes mRNA, miRNA, snRNA and snORNA genes. Okay. So that's why number C statement was false. So now I will move toward the next problem. Problem number 4 says, which of the following statements is not true regarding the chair cyclohexane? Your options are number A, the dihedral angle of 2 axial bonds of adjacent carbon is 180 degree. Number B, dihedral angle of 2 equatorial bonds of the adjacent carbons are 60 degree. Number C, the axial hydrogen atoms on C1, C3, C5 form an equilateral triangle, and number D, the dihedral angle between the axial bond and equatorial bond of adjacent carbon is 120 degree. Which of the following statement is true, is not true regarding the chair cyclohexanes? Means which of the statement is false? Read the options very carefully and let me know in the chat box. Let me check the chat box now. Number D, I think not two. I have only two answers which says it is the number D. Okay, let me check if it is the correct one or not. So the statement which is not true regarding the chair cyclohexane is number D. The dihedral angle between axial and equatorial bond on adjacent carbon is 120 degree. It is not true. Okay. So the dihedral angle is this. This is basically the angle between two planes which are intersecting each other. So in case of your cyclohexane, so what happens, like this is the chair cyclohexane, right? The chair cyclohexane, if you look at these two position, and this is the Newman projection of the chair cyclohexane, right? So this is the dihedral angle which changes, and also the sizes with the torsion angle. So the dihedral angle, so this is the axial bond, right, and this is the equatorial one. So what is the dihedral angle between the axial and equatorial one bond of a particular adjacent carbon? So what is the angle? As you can see, this is the angle between this and this, right? So the dihedral angle between two axial bonds are 180 degree. This is this and this. Okay, the angle between these two, the angle between this hydrogen and this hydrogen, this is the equatorial bonds is 60 degree, and the dihedral angle between this and this, it will be again 60 degree, right? We have 60 degree, we have then 180 degree, and then we have 60 degree and 180 degree. So this is the rotation wise. So the statement number D was false. So now I will go to the next problem. Problem number 5 says, which of the following is the main organ of drug excretion? Your options are number A lungs, number B liver, number C kidney, and number D sweat glands. Which of the following is the main organ for drug excretion? Lungs, liver, kidney, or sweat glands? Let me check the chat box. Number C, number C, number C, number C, number B. Okay, number C and number B is the most given answer. Let me check if number C is the correct one or number B. So the correct answer will be Yes, number C, kidney is the main organ for drug excretion. So, excretion is defined as a process whereby drugs and metabolites are irreversibly transferred from internal to external environment through the renal or non-renal route. Excretion along with metabolism and tissue distribution is important in determining both the duration and drug action and the rate of the drug elimination. Renal excretion, drugs are eliminated from the body primarily by the kidneys. The principal renal mechanisms are involved in the excretion of the drugs are glomerular filtration, active tubular secretion and active tubular reabsorption. The renal excretion of drugs, it is an irreversible transportation of drugs then they are metabolized from body to outer environment. So, this is the blood, this is the glomerulus and the proximal tubule, then this is the loop of the Henle and the distal tubule and the collateral tubule by which the drug goes to the urine. The first, the glomerular filtration happens for water, unbound drug and metabolites, then active tubular secretion for acidic basic drugs and metabolites, active reabsorption for acidic basic endogenous compound, passive reabsorption for lipophilic drug, and urinary excretion drugs and metabolites are that are filtered, actively secreted and non-reabsorbed. Rate of the excretion equal to rate of the filtration plus rate of the secretion minus rate of the reabsorption. Okay. So, this was the problem number 5. Now, I will go to the next problem. So, problem number 6 says which of the following natural products is used as an anti-malarial drug. Your options are number A Taxol, number B artemisinin, number C discopyrrole and number D curcumin. Which of the following natural product is used as anti-malarial drug? Taxol, artemisinin, discopyrrole or curcumin? Let me check the chat box. Number B, number B, number B, number B. So, number B is the most given answer. Let me check if it is correct or not. So, the correct answer will be yes, number B, artemisinin is a natural product that is used as antimalarial drug. So, this is the structure of the artemisinin. Okay, this is the structure. It has a this O ring, and this is a particular, this is the this is the reason of the particular activity of the drug. So, this is a particular artemisinia annula L. This is the natural product from which the artemisinin is synthesis, and artemisinin has several derivative like dihydroartemisinin, artemether, artesunate, etc. So, the mechanism of action, artemisinin and the prodrugs that are biologically active metabolite dihydroartemisinin, which is active during the stage of the parasite located inside the red blood cell. Although there are no consensus regarding the mechanism through which artemisinin derivatives kill the parasites, Several lines of evidence indicate that artemisinin exert their anti-malarial action by radical formation that depends on the endoperoxide bridge. This is the endoperoxide bridge. When the parasite that causes malaria infects the red blood cell, it consumes the hemoglobin within its digestive vacuole and a process generates the oxidative stress. These compounds have the presence of endoperoxide bridge which interacts with the heme of the parasite. Heme iron cleaves the endoperoxide bridge, and this generation is highly radioactive free radical which damage the parasite membrane by covalently binding to the membrane. Okay. So, this is the mechanism of action of artemisinin. And this is a particular your Chinese traditional medicine which also got the Nobel Prize in I think the in medicine in 2000 something, I think 2017 or 18 it got the Nobel Prize. Okay. So, now problem number 7, it says which strategy of drug design was used to develop the prometholol from the adrenaline. Your options are number A ring-fusion, number B simplification, number C rigidification, number D chain extension. This is structure is adrenaline, this is the prometholol. So, what is the drug design strategy was used to develop this drug from adrenaline? Let me check the chat box. Number C, number A, number C. Okay. So, number C and number A are the 50-50 given answer. Let me check which of the following will be the correct one. So, the correct answer will be number A, ring fusion. So, ring fusion was the drug discovery or drug design strategy that was used to develop prometholol from the adrenaline. So, the ring variations, for example, prometholol is the beta blocker. So, this is the adrenaline from which the particular prometholol is synthesized. It is selective for the beta receptors over the alpha receptors. So, beta blockers are beta adrenergic agents or beta agonists, or these are the class of the drugs that are particularly used for management of cardiac arrhythmias, protecting heart from second heart attack and after first heart attack, and also the hypertension. Okay. So, ring fusion is, this is the particular here, this is the, you know, we have a hydroxyl groups here from an adrenaline, but the fused system is introduced here in case of the propranolol, which makes this compound more selective toward the beta receptors. Okay. So, now problem number 8, it says that the use of one drug for activating different targets is called number A additive therapy, number B combinatorial therapy, number C empty therapy, and number D none of the above. Using one drug for activating different targets is known as additive therapy, combinatorial therapy, empty therapy, or your none of the above. Use of one drug for activating different targets are called which kind of therapy? Let me check the chat box. Okay, number C is the given answer. Let me check if it is correct or not. So, the correct answer will be yes, number C, empty therapy. So, okay, sorry. So, basically the drug which activates two targets simultaneously to have an enhanced therapeutic effects are, this therapy is known as empty therapy, which is the multiple target therapy. So, what are the importance of this therapy? Like the more predictable and superior pharmacokinetic and safety profiles as the consequence of administering a single drug. The combination of multiple active principles may result in positive and but also negative synergistic effects, which limits the number of useful combination. The probability of developing target based resistance simultaneously to multi-target drugs is statistically lower than the probability of developing resistance against single target drugs. The risk of the drug-drug interaction associated with the combination therapies is avoided and the therapeutic regimen is greatly simplified. The use of drug cocktails often complicates the dosing schedules and negatively impacts the patient compliance. It is more difficult to perform clinical trials with drug combination if drugs come to different companies and higher efficacy against complex diseases such as cancer. So, that is the advantages of the empty therapies. Okay. Now, problem number 9, it says which of the following terms refer to the molecular modeling computational method that uses equation obeying the laws of classical physics. The options are number A molecular mechanics, number B molecular, so number A quantum mechanics, number B molecular mechanics, number C molecular calculations, and number D quantum theory. So, which of the following terms refers to the molecular modeling computational method that uses equation obeying the laws of classical physics?
There's
Okay, the questions, I do not set the questions, okay, that will be done by others, the faculty members, that will be done. So, I do not know what type of question they will set in the examination. So, that is completely is not my area, okay. So, I cannot tell you, but I think it will be from the basic questions only, okay. Mostly it will not be any kind of very complicated question in the examination. So, you do not have to worry about it. Let me check the chat box. Number B is the given answer. Okay, let me check if number B is the correct one or not. Okay, so the correct answer will be number B, molecular mechanics. So, what is molecular mechanics? It is a computational method that computes the potential energy surface for a particular arrangement of atoms using potential functions that are derived using classical physics. Molecular mechanics force field methods ignores the electronic motions and calculate the potential energy of a system as a function of nuclear position only. So, the differences between quantum mechanics and molecular mechanics are like quantum mechanics, the classical laws fail at a quantum limit. Now, where does the electrons go? What does the electron do? Solves the Schrodinger equations like H equal to E psi, accounts for the explicit interaction of interaction and highly accurate. Whereas the molecular mechanics solves for the Newtonian equations of motion, F equal to ma, the atoms equal to are taken as a balls and bonds as springs, no electrons and much faster compared to the quantum mechanical method. The scales of the quantum mechanics is 10 to the power 4, 10 to the power 5. 100 atom is equal to 1 hour and 200 atoms in 16 to 32 hours, and scales is 10 to the power 1 to 10 to the power 2, 10,000 atoms in 1 second, and 20,000 methods can be given as 2 to 4 seconds. Okay, that is the scales of the quantum and molecular mechanics. Now, the last question of today's session in our course, it is in which of the following process the protein 3D structure of a target is a must requirement. Your options are number A, ligand based drug discovery, number B, structure based drug discovery, number C, fragment based drug discovery, and number D, all of the above. In which of the following process the protein 3D structure target is must requirement? In which of the following process the protein 3D structure of a target is a must requirement? Let me check the chat box now. Number B, number A, number D, number D. Okay, so I have A, B and D, these three different answers. Now let me check. So the correct answer will be number B, SBDD or structure based drug discovery. In this process, protein 3D structure of the target is a must requirement. So there are different techniques of drug design, which are the de novo drug design techniques. So are structure based drug design and ligand based drug design are the main. In the structure based drug design, receptor 3D structure is known and generation of interaction sites of the ligand can be done. In ligand based drug design, receptor 3D structure is unknown and generation of a pseudo receptor on directly similarity search. Then it can also, we have the atom based and fragment based drug design, and as in atom based, a seed atom is a requirement and higher the number of generated structure and the questionable chemical accessibility. In fragment based, a fragment is required, lower the number of generated structure with a good chemical accessibility. So what is the workflow of a SBDD project? The crystal structure of a protein available from literature. Then we go through the high throughput virtual screening to identify the hits and synthesize and purchase the hit, assay the hits and identify the active compound, and then we have the lead optimization. So this is the workflow of a SBDD or a structure based drug discovery program. Okay. So remember, 3D structures of protein is a must requirement in case of the SBDD project. So this was the last question of our today's session and also for our course. So we have successfully completed our 12-week live session for our medicinal chemistry course. All of you have responded very well in the question answer sessions and also have joined regularly. Few of you have joined very regularly, which is very I am very grateful. So one more thing, I will just show you once where to find the problem solving sessions and also the feedback form for the last time, because some of you have a little bit confused. So what you have to do is you have to go to the SWAM portal at first. Let me show you where to find all the lectures of problem solving session and recording, the recordings as well as the your presentation links. Let me show you, just wait for some time. Okay, 1 second. So first you have to go to the SWAM website. Here you will find, whenever you log into SWAM website, you will find this particular medicinal chemistry where you have all the week's videos and lectures. Here we have the problem solving session. If you click, you will have problem solving session recording. If you click on that recording, you will have this pop-up which says click here. If you click, you will have a sheet which contains the YouTube links of every class. Okay, if you click on the links, you will find the videos for each and every week, also the presentations. Okay, so you can check it from here, and also there is a your particular survey. Okay, this is a feedback form. So if you get little bit of help from the particular course, you can just fill this feedback form, okay, and submit. Okay, it will help me for improving myself for the TA-ship duties. And yeah, the Sunday I have already uploaded the Sunday house session PDF in that particular Google sheet, which is this one. Here it is a summary session. It has the presentation folder link. If you click on the link, you will find the particular Sunday's problem solving session. Okay. So now that for last, I am sharing my email ID. If you have any doubt regarding any class or anything, and you can just feel free to let me know. This is my email ID. If you have any problem or any query, it is still although our session is over, our classes are over, but still you can reach me through this email before your examination if you have any problem or any doubt. And also I will suggest you to download this book which I have written here, the Medicinal Chemistry book by Graham L. Patrick. You can just read this book because it has a very thorough discussion of the topics and in very easy language. Okay, so I think reading this book will also help you for your course. So now I thank you all for joining and keeping it up with the last 12 weeks. We have successfully completed our medicinal chemistry course, and all the best for your upcoming examinations. I know all of you can do very well. So if you have any problem, final test questions, I don't think we have we have the access to give you that. Okay, only we can give you some previous assignment materials, but we cannot give you the examination test material. Okay, still you can access it on the SWAM portal. There is a portion which is known as the your I mean the portal like this, which has a discussion forum. You can just ask there also. If it can be given, it will be provided to you. So thank you so much all of you. Please provide the feedback if you were, if the sessions were helpful to you, or if you have any doubts or any query, and I will close the session for today, and all the best for your upcoming examinations. So I am ending the session for today. Okay, thank you all.